Oh, hey. Morning. Today this is AP Bio Review video number 11. And between videos 11 and 12, we'll be looking at the cell cycle and a little bit more on cell communication. This one will focus on cell cycle control, proto-oncogenes, tumor suppressors, apoptosis or apoptosis, however you'd like to say it. CDKs, cyclins, stuff like that. To start off with though, well, first of all, I do have a projector running and the AC is on, so I know it's a little bit louder. The reason I don't normally use the projector is because it's kind of loud, but I tested it, seems like the audio is okay, so we'll give it a shot. To start off with, you might remember I've shown this video to some of my classes. In this video, obviously it starts off as just an animation, but in this animation, this is supposed to be a monolayer of skin cells on a microscope slide. And then what they're going to do is they're going to take a needle and scratch through them. This is the equivalent of you getting like a scratch on your arm. And so now these cells on either side have to become aware that all these cells are dead and gone. And so these guys have to somehow know to grow. And they're supposed to grow, grow, grow. Eventually, once they meet each other and they make contact, it's a phenomenon called contact inhibition. The fact that they contact each other inhibits them from growing any further, and then they're supposed to stop growing. If instead they grew and grew and grew and grew and grew, then even after they ran into each other, like right here, it'd start to pile up on each other, and that would focus into a small point, and that's what we call cancer. So they're supposed to grow and grow and grow and stop, but if things are growing out of control, that's cancer. The opposite of cancer is necrosis. So when cells are dying and they're not supposed to, that's necrosis. So those two terms will come up again, but that's kind of what you're seeing. So there's kind of two main parts to this video. How did these cells communicate with each other externally to know, hey, there's a hole there, we need to fill it? And then, how do cells go through the cell cycle? You might remember G1, S, G2, and then mitosis, prophase, metaphase, anaphase, telophase, and then cytokinesis. So today what we're focusing on is the cell cycle part, and then the next video will focus a little bit more on, like, once there's a wound, how did these cells talk to each other to basically heal the wound. So we'll begin with this picture, a familiar picture. And in the cell cycle, there really is no beginning and end, so to speak, but the logical place to begin is right after cytokinesis. Cyto is cell, kinesis is movement. So I don't know, cytolysis might have been a more appropriate term in my opinion, although maybe that means, like it exploded accidentally. This is intentional. So right here, we just made one cell just split into two, baby cells. So this first phase, gap 1, that's gap 2. Even though it stands for gap 1 officially, I think of it as growth. So these baby cells, so to speak, are now going to just physically become larger. Much like a balloon, if you just keep adding more air to it, or a water balloon, you keep adding more water, it just gets physically larger. And then eventually, if I'm going to divide again, essentially I need to expand the size of the cell, copy all the crap inside, and then divide. The analogy that I've given is if I wanted to make an exact duplicate of this room, then I would basically, take that wall and start moving it out, right? The room would have to get twice as big. And then I'd have to copy all the stuff inside. Then I'd just build a new wall, and now I'd have two identical rooms. That's kind of the logic. So the cell gets physically larger, then I copy the DNA. So that's the S phase, because we're synthesizing DNA. We refer to it as DNA replication typically in textbooks, but it's DNA synthesis in the cell cycle. So the cell got larger, we copied DNA, now we copy all the other crap. So organelles and such. I'm going to need more ER and Golgi and whatnot. And so that's what I'm copying right here in gap 2. Together, G1, S, and G2 are all referred to as interphase. But more specifically, interphase is G1, S, and G2. So College Board might be kind of broad, or they might be really specific with these. Then we get into mitosis. Depending on the textbook you want to use, you can break down mitosis into 4 or 5 stages. I tend to go with 4. So prophase, metaphase, anaphase, telophase, PMAT. A lot of books will say prophase, pro-metaphase, metaphase, anaphase, telophase. That's fine if you want to do that. So to me, having a phase called pro-meta is like, why wouldn't you just name it? Why wouldn't it have its own name if there's going to be 5 phases? I don't know. Seems sort of odd. So in mitosis, we are now going to go through some phases that look like this. So back in interphase, at the end of interphase, remember G1, S, and then G2 is the end of interphase. This is sort of what the cell looks like. So inside, my DNA, even though it's not quite as like just loose as it looks maybe in this picture, the DNA is fairly kind of strewn about. Not strewn about so much that it's getting tangled up in knots, but it's fairly strewn about. And so if you just logically think about, if I'm going to eventually have to take these chromosomes, and in mitosis I'm trying to separate DNA. So I've already copied it, because I copied it in the S phase, right? DNA synthesis. So if I've got this DNA and it's kind of strewn about, you know, the average human cell is supposed to have about 6 feet of DNA. The average adult human is trillions of cells, maybe up to 100 trillion cells. So if I have 6 feet times trillions of cells, I've got 6 trillions of feet. Or if I've got 10 trillion cells, it's 60 trillion. If I've got 100 trillion cells, 600 trillion feet of DNA. And so I have to cram all that in there without it getting tangled. I also have to be able to move it. So if I have the equivalent, if this room were a nucleus, then the amount of DNA that I would have inside would be trillions and trillions of feet. So let's say I've got a string that's 1 trillion feet long, and it's strewn throughout the entire room. And I want to move it from, you know, everywhere in the room to over in that corner. If I just grab a little piece in the middle of it, so here it is, if I just sort of like grab this part here and take it over there, I didn't get it all. It's still strewn throughout the room. But if I could pack it down, kind of coil it like this, maybe around some histone proteins, then now my DNA is sort of condensed into chromatin. Chromatin is an IN. IN means protein. That's because it's DNA wrapped around protein, histone proteins. So now I can move this easily. Again, if my DNA is strewn about everywhere, and let's say this is one side of the room, and I grab a part in the middle and move it, now I got to go back and get more and more and more. But if it's all packed down into chromosomes, now I can move it more easily. So in prophase, we have 3 things that are going to happen. Number 1, once we're in prophase, my DNA will be packing down into chromosomes. Some of this might start at the end of interphase. The cell doesn't decide, go, it's time for prophase. Everything's just kind of like, it's like a steady state. So yeah, at the end of interphase, you'll see the DNA condensing, possibly. So in prophase, the DNA condenses. Okay, well eventually, we want to line everybody up in the middle, metaphase, to separate them. So somebody has to like reach out and grab those chromosomes. So you might remember we had centrosomes. In animal cells, those centrosomes have centrioles in them, but apparently you don't need centrioles to work, for your centrosomes to work, because plants don't have centrioles. So this little starburst looking thing right here is a centrosome, and there's another one. We want 2 in a cell. We don't want 3 or 4. We'll see why in a second. So these are, basically the analogy is they're kind of like fishermen, and they're going to send out spindle fibers to grab the chromosomes and reel them apart, kind of like they're fishing for, you know, chromosomes, you know, like you do. So, back over here, in prophase, 3 things happen. 1, I have to condense down my DNA into chromosomes. 2, centrosomes appear. So around the end of interphase, centrosomes can just appear. And then they're going to start making spindle fibers. So that's the second thing. So centrosomes appear and they start making spindle fibers. DNA condenses, centrosomes appear with spindle fibers. But the problem is, you'll notice the centrosomes are on the outside of the nucleus. The chromosomes are on the inside of the nucleus. So those spindle fibers can't reach. So what do we have to do? We have to get rid of the nucleus. Hopefully we turn off any enzymes that might break down DNA, DNases, in the meantime. But the third thing is we have to dissolve the nuclear membrane, the nucleus. That way, these centrosomes can send out their spindle fibers, made of microtubules, to grab the chromosomes. Then what we want to do is line up these chromosomes in the middle and pull them apart. So back to the analogy from earlier in the year, the Halloween analogy. Let's say that I've got a giant pile of candy. My brother and I, we go trick-or-treating. We get back home, we dump it all in a pile, and now we want to break it up evenly and pretend that we happen to have, 2 of everything. So I want to somehow make sure that we get identical piles. So what I would do is I'd have like Snickers, I'd pair those up. Then I'd have like Milky Way, pair those up. Then I'd have Kit Kat and Kit Kat, pair those up. From there, if I just like basically cut this down the middle and divide everybody, I've only got so many hands, but if I just cut this down the middle and divide everybody, then now each pile is equal. That's what I'm trying to do in mitosis. So if that and that are identical, then if I pair them up, Snickers, Twix, Three Musketeers, Almond Joy, then when I pull them apart, that side and that side will be equal. So let's go back to this again. Drop in a little bit more vocab. In G1, in human cells, you should have 23 pairs of chromosomes, assuming you're normal. So in G1, I've got a chromosome 1 from dad and a chromosome 1 from mom. I've got a chromosome 2 from dad and 2 from mom, a 3 from dad, a 3 from mom. So all the ones from dad obviously came from sperm, and the ones from mom obviously came from her eggs. Don't think about it too much. Well, what we're going to do is we're going to simplify it and say that, pretend that we only have 1 pair of chromosomes. I get a 1 from dad and a 1 from mom. They're not identical, because mom and dad are not identical. Hopefully, that'd be impossible. So as I'm going through the G1 phase, this is what I look like. These chromosomes are called homologs, or homologous chromosomes, if you want the adjective version. So as I go through G1, I've got a 1 from dad and a 1 from mom. When I go through the S phase, I now make copies, DNA synthesis. These copies are called sister chromatids. You might hear them called chromatids for short. So that and that are identical, assuming we copy them perfectly, which we will have some errors probably. That and that are also identical. Again, our error rate on DNA replication is about 1 in a billion base pairs. Our genome is a little bit over 3 billion base pairs, like somewhere around 3.1, so we'll pick up a few errors, but these are supposed to be identical. So blue to red, homologous. Blue to blue, they're identical, they're sister chromatids. And they're glued together at a centromere. That's important, because that's what's pairing them up. Remember back to my Halloween analogy? Snickers, I pair them up. Milky Way, I pair them up and line them up in the middle. So when I make identical copies, sister chromatids, I join them together at centromeres. Join these together at centromeres. Now they're paired up. So if I can just line them up and then pull them apart, we'll be equal. Terrific. So back down here, these guys are already paired up because that happened in the S phase, right? So prophase, DNA's condensed, spindles and centrosomes appear, nucleus dissolves. Middle, metaphase. Everybody lines up in the middle on metaphase. These spindle fibers will attach to these special proteins called kinetochores with a K. And I'm assuming kinetic, movement, kinetochore. And the spindle fibers will attach to the kinetochores and then pull these apart. Anaphase, apart. Metaphase, middle, anaphase, apart. Now, once these guys are moving apart, this is anaphase. Once we're no longer moving, then now we're done with anaphase. And then the last one is telophase. Well, telophase is just going to be the opposite of prophase, which is logical. If you think about it, back in prophase, why did we condense DNA? So that we could eventually pull it apart. Why did we dissolve the nucleus? So that we could eventually access the DNA and pull it apart. Why did spindles and centrosomes appear? So that we could pull DNA apart. Okay, well now, we've done that. We're done pulling it apart, pulling DNA apart, the chromosomes. So now in telophase, just put everything back like you found it, the opposite of prophase. So now, once we've separated everybody, do we need centrosomes and spindles anymore? No. So in telophase, centrosomes will go away, spindle fibers go away, they dissolve. Now we're going to need nuclei, so we'll start to build a nucleus and another nucleus, opposite of prophase. And then lastly, when my DNA is super condensed down, there we go. When it's super condensed down, it's tougher to read those genes. Those genes are off. So now my DNA can relax and I can start reading genes again. So you'll notice the DNA starts to kind of uncoil a bit right there. Okay, we can see this. Let me show you kind of a video of it. And so in this video, my DNA is already condensed into chromosomes. That's why it looks like snakes. I can kind of see the outline. It looks like everybody's encased in this egg-like structure. And right about now, it looks like everybody, apparently the nucleus just dissolved because now everybody's a little bit more kind of free, so to speak. So we're in prophase for sure now. Starting to look like everybody's possibly getting lined up along the middle here somewhat, maybe about like that. So we might be in metaphase now. Yeah, starting to line up. So anaphase, anaphase. Notice my DNA is still condensed. It still looks like worms. Anaphase, anaphase. Now watch my chromosomes right here. They look like worms. They look like worms. They're condensed. They're now uncondensing. They're relaxing. So now it looks more like a cloud. And then we'll build a nucleus around each one. This one happens to be a plant cell, so they're going to build this thing in the middle between them called the cell plate. That becomes the cell wall. I wouldn't sweat it too much. So plant cell division looks a lot like animal cell division, except not only do they do PMAT and then cytokinesis where the cell pinches in half, they also have to build a cell wall. But other than that, it's pretty much the same. Here's some stained pictures. There I can see the chromosomes are pretty condensed, so that's probably prophase. I can see the red spindle fibers, the red stained spindle fibers there, so that's probably prophase. Kind of looks to me like the nucleus might not be totally dissolved yet, but 2 out of 3, I'm calling it prophase. There everybody's lined up in the middle, metaphase. Here everybody's pulled apart, anaphase. And here I'm starting to build a nucleus and a nucleus. You'll also notice that the DNA is relaxing. It doesn't look like snakes or worms anymore. It just looks like a little bit more cloudy. So that's telophase. And you can see right in here, it looks like pretty soon the cell's gonna start to do cytokinesis and finally split. So technically, mitosis is separating the genetic material, the DNA, and then cytokinesis is splitting the cell. Cytokinesis is technically not part of mitosis. Although what's kind of confusing is they'll call the M phase mitosis and cytokinesis. So I don't know. If mitosis and cytokinesis are going to be different, I don't think that you should call them together the mitotic phase because that's confusing. So here's a quick picture of what sister chromatids look like. Remember, in G1, I have a chromosome 1 from dad, chromosome 1 from mom. As I go through the S phase, I make copies. And so those copies will be together. That's why when you see chromosomes, you usually see them as X's, because you usually see them after the S phase, after they're condensed. In like say prophase or metaphase or something like that, those are the pictures you always see. So that's why they always look like X's. And you can see how they're joined together there at the middle, at the centromere. So centrosomes make spindle fibers, centromeres join the center of sister chromatids. All right. There you go, just for fun. We're in the middle of metaphase. Anaphase, still condensed, and watch the green if you can see it. And it just went uncondensed. It sort of changed in color there. If I go back and look, see how it's less, that's where it's more condensed. That's where the DNA is less condensed. All right, so the next part that we want to focus on, and this is actually what is the stuff that we just mentioned, is fair game on the AP Bio test. But what's more likely and what is more emphasized in the current College Board curriculum is how we control all this. So cell cycle control. So we've got a few checkpoints. I'm not that concerned, and I doubt College Board is that concerned with exactly where is this checkpoint, but the idea is this. If I've got a checkpoint somewhere between G2 and M, what am I checking for? I'm probably checking for making sure everything else was done since the last checkpoint. That's what logic would tell me. So it's making sure that basically we've copied our chromosomes and stuff like that. What would be horrible is if we're only halfway through the S phase, we're duplicating organelles, so we've only copied half of our DNA, and then we go ahead and start doing mitosis. That would be a wreck. So essentially right here, we're trying to make sure, like, hey, does everybody have their partner? Like, you know, do I have my sister chromatids, and are they lined up? That type of thing. So that's what we're doing at those checkpoints. Well, how exactly does a cell do this? Because that's just a rectangle. Let's zoom in on this checkpoint, because checkpoints can be kind of messy. So all of these little blobs here are proteins. And don't worry about the color coding. There's a chart for the color coding. You know, if you're, just to show you that there is one. If you're that color red, you're a kinase. That means you add phosphates to stuff. If you're that color green, then you're a phosphatase. That's where you take phosphates away. If you're blue, you're a transcription factor. So there is a color coding and a shape scheme. Like some of those, that looks like kind of a hexagon. So back up to this picture. This is cell cycle control. So what you'll notice on all of these is right there that says G1 phase and that says S phase. So this is sort of like the checkpoint. Well, if I sort of look at the diagram, all these proteins that are passing along information, they all feed down into these two spots. That kind of red thing right there is CDK. That's another CDK. Don't worry about that one's CDK2 and that one's CDK4 and 6. But these CDKs are the ones that say proceed. They are activated by cyclins. So essentially what it looks like is I've got this CDK, a cyclin-dependent kinase. Kinase, like I said a second ago, are enzymes that add phosphates to things. A lot of times, when something becomes activated, it gets activated by being phosphorylated. So here's a protein, and it's off. I add a phosphate by burning an ATP, and now it's on, and it'll go do its job. So you might remember from videos 9 and 10, I could have a chain of proteins. This protein's supposed to turn on this one, supposed to turn on this one. So it could be this guy gets phosphorylated, okay? Now he'll phosphorylate this guy. Now he's on. And he'll phosphorylate this guy. Now he's on. And he'll go like turn on a gene. Maybe he's a transcription factor. So he'll help turn on a gene. Okay, so back to this. I've got these CDKs. This is a cyclin-dependent kinase, so it obviously depends on cyclin. And the job of cyclin is to turn this enzyme on. So it's born, so to speak, in the off position. The CDKs are always present in the cell. So here it is, and it looks like this. So right now, with this shape, it can't fit its substrate. There you go. I was never very good at puzzles, apparently. So it can't fit its substrate. But if cyclin comes into the mix and binds to an allosteric site, instead of acting like an inhibitor from earlier videos that acts as an activator, now the shape changes. And now, if you look at it from the side, now I do have a good fit. And so now I turned this CDK on with cyclin. So what's turning this CDK on? That's cyclin. This one happens to be cyclin D, whatever. What turns that CDK on? This cyclin, cyclin E. So I kind of think of it like, I tried to do a government analogy earlier in the year and you guys made fun of me. I'm going to try it again. I've got two CDKs, so I'm going to think of the Senate, because you get two senators in your state. And so these guys basically get activated by these guys. So these are sort of like the main people that have influence over them. They're buds. And they're going to say, all right, let's go on to the next phase. All right, cool. We're going to come back to these guys in just a second. Let's look at another one. If I go back and I look at this phase, the G2 to M phase checkpoint. Now we see something like this. Now I don't know that you can actually read that from where you're at, but that says CDC2. That's the same thing as a CDK. So sometimes they'll discover these in like yeast or something and they'll name it CDC. And then in humans it's called a CDK. but they're interchangeable. So the CDC2 is the same as human CDK1 or 5 or whatever the heck. So it still works the same way. Again, here's my CDK. I'm going to go ahead and call it a CDK. It's activated by cyclin, and if that's true, then we go from G2 into the M phase. He says, proceed, you pass quality control. All right, that's great. That seems simple enough. Let's look at a graph real quick, and then we're going to come back and explain all these other guys. So in this graph, if you can't read it very well, that says G1 phase, S, G2, mitosis. This is cyclin D. So this red line is cyclin D. Cyclin D is always at decent levels. I realize it's not at a high level here or here, but it looks like cyclin D is going to be around always. That just means like, hey, we're cruising through the cell cycle. Maybe the absence of cyclin D is what kicks you into G0. G0 is when you get out of the cell division game. So back up here, instead of just always going towards dividing more and more and more, I can exit the cell cycle out of here, out of G1, and go into G0. G0 just means you're not growing anymore, you just do your thing. So for example, brain cells go into G0, and as a general rule, they never leave. That's why you can't grow new brain cells. So they're still doing their job, they're still carrying electricity, nerve impulses, but they're just out of the game of growing. Cardiac cells in general go into G0 and don't really come out. Skin cells, they're always going through the cell cycle because I'm shedding skin cells all the time, so I need to constantly grow new ones. Okay, so back to our graph. So cyclin D maybe is required to not be in like G0, or to, yeah, not be in G0. Cyclin E, the green one right here, it looks like that one crests, and that's what sends you into the S phase. In G2, it looks like cyclin A, this blue one, is getting really, really high. That sends you to the next phase. Cyclin B, the orange guy, it apexes or crests up here, and so now we're ready for mitosis. So the point is this. The CDKs are always around, but they're inactive. Until, if you're ready to go on to the S phase, then you'll make a bunch of cyclin E, for example, that will bind to the allosteric site. This active site will change shape, and now it's on, and now we start going through the next phase. Okay, now what we want to do is look at everybody else. So back to my government analogy. This is the senator, and maybe, you know, somebody really close to the senator. Maybe if the senator's a woman and she's married, maybe that's her husband, and he has strong opinions about things too. So he has a strong influence on her. These are the lobbyists. So maybe NRA has a lobby, or some sort of teachers' unions. We don't have teacher unions in Texas. But, you know, we have all these different groups. And so they are trying to influence the senator. So when I look at these, you'll notice, I'll zoom in a bit, some of them are arrows, and some of them are what I just call T-bars. An arrow means go. A T-bar means no, it means stop. So what we want to do is we want to kind of trace the pathway. And earlier in the year, I said for some of you guys, it'll be easier to trace the pathway from the top down. Others, it'll be able to trace the pathway backwards. So, for example, CDC 25. Let's zoom in. It tells this CDK, again, I know it says CDC2, but we're calling it a CDK. It is sending an arrow to CDK, so it's saying proceed. He's basically sitting here going, I want to move forward. Cyclin says, you should move forward. But he is also agreeing, yes, move forward. I support this idea. He says, I want to pass this bill. Cyclin says, pass the bill. And he's like, yeah, I agree, you should pass that bill. CDK7 says, proceed. Then I start getting into some of these. Protein 21, P21 is saying, no, don't do that. Don't pass that bill. They're having an influence on this. So cyclins and CDKs, it might be like, hey, I'm 10 years old, it's time for me to get taller. So cyclins are produced based off of time. But all these other proteins are influencing, like, do you have enough calcium? If your bones are going to get taller, you need plenty of calcium. Let's say you're intaking very little calcium. Do you have enough calories in general? Let's say you're only getting 400 calories a day. You probably don't want to like grow. You want to try to just like not die. So these guys are putting in influences. Let's back up a little bit. This will hang with me for a couple minutes and it will make sense. So here with CDC25, he says go. Here's check 1, CHK1. Check 1 tells him no. So this guy is overall stopping the process. CDC25 is overall supporting the process. This guy is saying go. So he is what we call a proto-oncogene. Things that in the end, their final, whenever you map them through, if their final signal or their final influence is go, go, go ahead and divide, that's a proto-oncogene. A proto-oncogene is something that could potentially, when mutated, and it's locked in the on position, is now an oncogene. Onco means cancer. So when cells are growing out of control, that's onco, cancer. All right, why would this guy grow out of control? Well, maybe if he's always saying go, 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 then he's like, all right, cool. So the cell grows out of control. This guy stops him. So this guy right here says grow, but this guy says stop it. So he is what we call a tumor suppressor. His overall effect in the end is stopping the process. So now let's look at how this would actually work. And this is where hopefully it all makes sense. Let's say I lost some cells, so it's time to grow some more skin cells. All right, so cyclins-cdks might be saying, let's go. But what if I've been out in the sun and my DNA was damaged? I wasn't wearing sunscreen and, I'm white, if you haven't noticed. And so now that UV light can damage my DNA, potentially causing cancer. And so now if I divide, I'm basically copying the cancer, right? If UV wrecks my DNA, and then I go ahead and go through the cell cycle and I make a copy of that cell, that's 2, and then copies of those, that's 4. I'm now copying the errors. So what if UV light damages my DNA? Or infrared light damages my DNA? So now I would activate this protein ATM, cool, which will activate P53. And then P53 activates that guy and that guy and that guy, for example. And what do those guys do? They go down and say, stop it, stop it, stop it. So what they did is they're basically saying stop. Why? Your DNA is damaged. Don't make copies. Back to my analogy earlier. You can also have pathways that proteins become active whenever there's like not enough calcium or not enough calories or something like that. So that could inhibit your growth. That could stunt your growth. Maybe there's a kid and his genetic potential is he'll be 5'10", but he's malnourished. So he doesn't get to 5'10". He's 5'8", or 5'6", or 5'4". So that's actually what's happening behind the scenes. So CDKs are activated by cyclins and they say go forward. But tumor suppressors overall eventually say stop it. Proto-oncogenes overall eventually say go. So P53, for example, is the most studied, most well-known tumor suppressor. And you'll notice he activates this guy. You're like, wait, he's not a suppressor, he's saying go. Right, but he's telling this guy to go, and this guy is saying stop. So this is basically me telling my friend, hey, tell CDK to stop. And my friend's like, okay, hey, you stop. So P53. You'll notice that P53 tumor suppressor is kind of a boss, so to speak, bossing around a lot of other proteins here. But you'll notice that P53 is in multiple pathways. If I go back and I look at there it is. There's P53, is in multiple pathways. So it's the interplay between growth factors and proto-oncogenes that determine whether or not you get to go on. This is the type of thing that you're more likely to see on the AP Bio test. And the way that they might ask it is, I doubt that they're going to care if you know the terms proto-oncogene and tumor suppressor. They might use them. If they pick a tumor suppressor to talk about, it's probably going to be P53. Common proto-oncogenes would be like RAS, there's BRCA, that's the breast cancer gene, although you'd think BRCA is because it causes breast cancer. It was actually discovered at Berkeley, California, and then it just happened to be breast cancer. So anyway, they'll probably give you, but what they would do is they might say like, what would happen, is the cell more likely to divide or less likely to divide if CDC is broken? Like CDC is mutated and it doesn't work. Well, the job of CDC is to say go. So when there's less of this CDC25, then that would mean no. That would mean that I don't grow as well. What would happen if P53 is mutated and it doesn't work? Well, P53 normally says stop, 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 stop. So now the cell is more likely to grow out of control. The cell is more likely to have cancer. Again, growing out of control is cancer. Dying when you're not supposed to, cells dying when you're not supposed to, is necrosis. Another thing that kind of comes up with this is whenever cells realize, and we'll talk about how in the future possibly, it might be the immune unit that's kind of an extra unit. But when a cell realizes that something is wrong, or an immune system tells it that something is wrong, then it can commit programmed cell suicide, apoptosis. Let me check my time really quick. Yeah, okay. Apoptosis. And in apoptosis, basically in all nucleated cells, which is everything except red blood cells, when red blood cells mature, since red blood cells are just containers of hemoglobin, and the job of hemoglobin is to carry oxygen, if you have a bunch of mitochondria in there, those mitochondria are going to eat up oxygen through cellular respiration. Respiration is breathing in oxygen. So the cell ejects its mitochondria, and nucleus and everything else, they're basically just containers of hemoglobin. So all other cells, though, in their DNA, there's a genetic code for cell suicide, apoptosis or apoptosis. It's just that those genes are off. But if this cell has some sort of damage, again, like if there's UV damage and we can't get in, that says right there DNA repair, then we can stop the cell from dividing. We'll try to fix it. If we can't fix it, then we'll just tell the cell to kill itself, which at first seems bizarre, but I have cells die all the time. So if I have a cell that's got a messed up genetic code, messed up software, so to speak, inside of it, you just tell that cell, hey, kill yourself, and the cell's like, all right. And you just take one of the healthy cells and make copies of those. So apoptosis is a way that we avoid cancer. And so it's normal for our cells to do that. So cells growing out of control, forming giant lumps of cells called tumors, that's cancer. Necrosis is cells dying when they're not supposed to. Apoptosis, is when cells are killing themselves on purpose. So if they're doing it in the correct situation, then that's just life as it is. If I'm at the Expo pen making factory and 1 out of 1,000 Expo pens comes out really, really messed up, I don't package that because now that ruins my Expo pen reputation. So I just take the bad ones and I throw them away. So don't you need 1,000 pens? You know, if 1 out of 1,000 is messed up, then now you only have 999. Well, I'll make 1,001 and then I throw one away. If I throw away 5 of them, then okay, I'll make 1,005 and throw 5 of them away. It's fine. So I kill this cell, all right, fine. I'll just grow another one, it's cool. The last thing that we want to mention about cancer is when cells are growing out of control, one of the limits, we'll see this in probably 2 videos from now when we do DNA replication, probably around video number 13, is we have these things on the ends of chromosomes called telomeres, because every single time that cells divide, the DNA gets a little bit shorter. Again, we'll look at why on, it'll probably be video 13. It might be video 12, but probably video 13. And whenever we do DNA replication, so you can look in the descriptions. And so one of the things with cancer cells is we keep rebuilding the telomeres on the ends of chromosomes. So cancer cells are relatively immortal as far as being able to divide forever. The rest of our cells, they divide, the DNA is shrinking and shrinking and shrinking. Once the telomeres get short enough, the cells can't divide anymore. They call it like their Hayflick limit or something like that. As far as free response that relate to this topic, 2015 number 4 is, it's a pretty good question. It's not covering the main things that I think would be on the AP Bio test. One of the previous 2 questions, I think it was like 2018 number 2, from our last video, one of those 2 questions is actually more like it. The question, here I can pull it up really quickly. I'll show you which one is more likely to kind of show up on the AP Bio test. This one, yeah. 2018, number 2. So in here, when they're looking at like, that's an arrow, so this guy activates this guy or turns this guy on or off or whatever. This one is a little bit more like what the cell cycle stuff would look like. Or cell cycle, cell communication stuff. All right, that's it for this video. Our next video, we'll look at kind of wound healing and cell communication. It's actually kind of the thing that I'm pulling out of the immune unit, but it's really interesting. It'll explain why we get a fever, why histamines, why antihistamines would work. Obviously, they're the opposite of, they're inhibiting histamines. We'll look at that. So, thanks.